The trick that you use is that putting these pieces back, like assembling them together like a jigsaw puzzle is a difficult task. It's computationally quite challenging. Instead, you use one person's human sequence, let's say the blue sequence. Take one person's genome sequence, and use that as a rough scaffold to say, since two humans are very similar to each other, if I have a little piece that I get from my sequence, it's likely to be present in the so-called reference sequence, somebody else's sequence as well. So I search for this piece in the reference sequence, and then wherever I find the hit, I place it at that location. So when I take each of these small pieces that I get from my genome sequence, if I'm sequencing mine, and I place it at the right position, then the difference between my genome and the other person's genome, the blue one, become apparent. If all of these pieces are saying there's a T here while the other guy's genome is saying an A, then it means I have a T while the other person has an A. And just by way of terminology, these things are called reads, these little strips. And that character is called a variant character. I'm different from the other person. The other person is sort of representative of a healthy sequence. It's not a single person, let's say. So imagine, there's no such thing, but imagine there's a healthy genome sequence, whatever that means. And so now that variant character T is potentially a character. If I had a particular disease, then maybe that variant character T is responsible for that disease, potentially. Now, not all variant characters will show up in all of the reads. So you could have a situation like this where half of them say there's an A and the other half says there's a T, because we have two copies of the genome, so one from each parent, so one of them might say something different from the other, and so you get situations like this. And all of that is important because we all know from Mendelian inheritance that some diseases are recessive, some are dominant, so sometimes one variant character suffices to cause the disease, sometimes you need two variant characters, and there's a whole gradation in between as well. Okay. And then the biggest challenge that one faces is, suppose I have a particular disease, I sequence my genome, and I get a list of these variant characters, either present in both of my genome copies or in one of my genome copies. Typically, that list is about 4 or 5 million characters long. There are 4 or 5 million characters that differentiate you and me. And now, how do I go down to the one that's causative of the problem that spurred my genome to be sequenced in the first place? Right? That's the basic challenge that we face. And a variety of techniques are used for that. A lot of knowledge has been built up about the various genes and what they do, etc. And so you have to bring it all together to do that funneling down to that one character. So the rest of the talk, I'm going to walk you through a collection of examples of various patients who've we've worked with and who've had various different sorts of indications, clinical indications. And in each case, we've had to run through this task of taking not quite 5 million. You actually for practicality reasons, you end up sequencing only certain sections of the genome, not the entire genome. So you get maybe 30, 40,000 variants as opposed to 5 million variants, still a large number. And then you have to distill down from those 30, 40,000 down to one. And how we did that, and what are the various phenomena in the genome that determine, how is it that a single character can cause a major change at a phenotypic level. Yeah.
So is it really one? Did you say one, or is it a combination of things?
No, one. So we only look at one because once we master one, we will look at combinations 10 years down the line. So today, one is a big enough challenge. By one, I mean it could be two, it could be one or two, right? Typically not more than that. Sometimes we should really talk about events as opposed to characters. So sometimes it could be a whole event, a whole chunk that goes away. It could be 100 characters, but it's one event, so to say. So a lot of it is just knowledge that's been built up through painstaking experimentation on various animals, and you knock out certain genes and say, okay, this gene is important, and then you say, okay, this character changes in this gene, can you predict what the impact of that character on that gene will be? And you put all of those pieces of knowledge together in some form. And there are various shades. There are genes about which nothing is known. So about 100, if 100 patients come to us, we are able to solve only 30% of the cases today. So the remaining 70% for want of lack of knowledge go unsolved. And why are they unsolved is all, we rack our brains and scratch our heads about what can we do about them. Okay. So So we talked about this, right? So if you have a single character change, then what does it do to a gene? We all know what the genetic triplet code is. The way recipes are written in the gene, there are triplets, 3 characters at a time. Each of this, each triplet stands for an amino acid. So in bold, you'll see the amino acid symbols. There are 20 of those. And if you have a variant, for instance, that you had the normal is a TTT and it becomes a TTA, then the amino acid changes from F to L, and that might change the protein dramatically, and so you might have major changes in effect and so on. Right? That's how things work. One more fact before I get to the cases. These recipes in these genes, there are about 20,000 genes in the genome, and each gene is a recipe, a collection of triplets which describes the protein. These recipes are not written out in one contiguous chunk, rather they are written out in interspersed chunks. So you have these so-called exons where the recipes occur, and then in between there are long stretches where nothing happens. So somehow our cells know how to take exon 1, jump all the way to exon 2, jump all the way to exon 3, and then concatenate all of those sequences, and then partition them into triplets and then convert it to proteins. So all of that machinery somehow exists in our cells. Okay. So what I want to walk through now is a sequence of cases, and each case there'll be a different sort of twist to the algorithm or something that you'll see, or a different sort of event in the genome, and building up with increasing complexity, while at the very end you'll see a fairly complex event going on. So, so here's a case where there was a family where there was a lot of individuals in that family who were going blind in their 30s and 40s. In the central part of the retina, the cells were dying. Okay? And that's called Stargardt disease. That's the name there. So when we sequenced the genome, this was the easiest case to solve, because when we sequenced the genome, we found variants in a particular gene that's well known to cause this disease. Right? So that's easy. So if you have a gene that's well characterized, it's very simple. And what happened was, now how does a single character lead to a problem? So here is a single character, the C changes to a T, and you have the triplets in this gene, and if you see what happens to the triplet, the R changes to an X. R is an amino acid, one of the 20 amino acid symbols, so it changes to an X. X is a special amino acid symbol, which means end of recipe, basically. So the recipe comes to an end out here, so prematurely. So one character change and the whole recipe comes to an end, so the protein is partial, and usually it's discarded when it's partial. So this gene doesn't happen, basically it's not created at all. Okay? Of course, that's in one copy of the genome. Remember, we have 2 copies. In the other copy of the genome, there's a more subtle event where a G changes to an A. So the amino acid changes from a G to an E. And that's a more subtle event. So what happens when in the protein, there's a one amino acid that changes into another. And that's very hard to predict, and we'll see why this creates a problem. But in this particular case, this amino acid has been well characterized. People have done experiments and shown that that causes the problem. So what is the problem that eventually happens? So this copy of the gene is gone. There's a premature truncation there, and now there's an X out here, and it's basically wiped off. Here this copy of the gene is still holding fort. It's a recessive disease, so one copy is good enough for you to manage. This is still holding the fort, but a change from amino acid G to E causes changes in the function of the protein. And what does the protein do? All it does is, when a light falls on the eye, there are a series of reactions, a lot of side effects happen, a lot of side products are created. Those products are toxic, and somebody has to clean these products out, so that's what this gene does. So in the absence of this copy of the gene completely gone, this copy heavily compromised, those products are not cleaned out. So over 30 years, they accumulate. And over 30 years, the cells start dying just because of toxicity. So it's amazing how just single character changes destabilize entire proteins, which serve very important functions, which lead to blindness over 30 years. Let's go to a next case, a little more complex. In a previous case, we saw that one character was replaced by another, and that happened for 2 different characters, and so that caused the problem. So here the picture is slightly more complex. In this case, the picture is that these 2 characters are gone from one copy of the genome, and that's what these 2 black spaces represent. The second copy seems to be intact, there's no problem. So one copy of the genome is intact, the other copy, 2 characters are gone. Okay? And this is a case of heart failure where 3 or 4 members in the family, by the age of 25, their heart had no power at all to pump. And you can see what happens to the triplets in this case. So if you have 2 characters that are gone from the recipe, and the recipe is made up of triplets which are synchronized every 3, then when anything but a multiple of 3 happens, these 2 characters go away, then the recipe completely jitters. And then you get a random sequence after that, pretty much. And more often than not, and you can do these calculations, more often than not, in a random sequence, you'll hit an X very soon. While in a true gene sequence, it waits for a while until the end of the recipe. So this recipe is also practically truncated, but for different reasons, because 2 characters are gone. And what it does is, this gene contributes to these rivets that connect heart cells together. So these rivets out here. So heart cells have to pump, right? They all have to pump in unison. So you have to have strong bonding mechanisms between neighboring cells so that they pump together rather than throb in place. And this gene contributes to these rivets. And if the gene is gone, then these rivets are compromised heavily. And so essentially the structural framework of the heart is compromised. So over 20, 30 years of pumping, it loses all its ability to continue to pump. Now the challenge in this case is that this happened in 1 copy of the gene, not in both copies of the gene. And that left you thinking whether can't the other copy of the gene compensate for that 1 copy that's problematic. And that raises lots of questions. In some cases, indeed, the other copy can compensate. Some cases it doesn't, and which cases does it, which cases it doesn't, is hard to say. For instance, so one of the things that we look at is take normal people and say, are there, along the length of the gene, are there such truncation events happening in normal people? So take 50,000 normal people, and such databases are available. And you'll see that a lot of truncations are seen at the end. So clearly, normal people seem to have lots of truncations at the end. So truncations at the end in 1 copy seem to be managed. Somehow, the protein is still intact. But truncations in the middle, there are very few instances of normal people, and these normal people also might develop diseases as they go along, etc. So this sort of tells you that truncations in 1 copy of the gene in the first, let's say 3/4 of the gene is problematic, but toward the end of the gene, it may be okay in 1 copy, right? It's just the length of the gene. And this signifies how many normal people have a truncation here, have a truncation here. So most of the places, the empties are all, there's no truncations at those places, right? So there are a few normal, maybe a handful, 7 or 8 normals out of 50,000 who've got a few truncations in between. There's a greater focus here because there are lots of normals with truncations there. So it suggests that truncations at the end of the gene don't seem to matter as much. People seem to carry along, but truncations in the middle are more problematic. But that's not true for all genes. So occasionally you'll find genes where this is a gene which plays a role in our muscle cells in the heart have this spring-like capability to expand and contract. And literally there's a spring in them. That spring comes from this gene. And normal people, there's a huge preponderance of truncations right in the middle. We don't know why. So somehow truncations right in the middle in 1 copy are tolerated, but truncations at the end are less so, etc. So there's still a lot of unknowns out here as to if you have 1 copy lost in a gene, does it cause disease, and whom does it cause disease, and so on. So those are all things we grapple with, yeah. No, protein structure doesn't matter because 1 copy of the gene is gone. Now, the other copy, does it hold fort or not? So is it capable of doing the job or not is one question. The second question is, when you have a truncated protein, it's not that it's gone. What happens to it is that cells have mechanisms to detect that the protein is truncated, and they actually destroy those proteins. Occasionally, the destruction mechanism fails, and so these truncated proteins escape and come into use. If they come into use, imagine those rivets, right? If half of the rivets are made from truncated proteins, clearly it won't work. So if they escape those destruction mechanisms and come into play, then they create a greater problem. So now which truncations escape those destruction mechanisms, which don't, and all of those are the questions that we don't know enough about. Okay? So then we run into these problems. So this is another case where 2 young kids who died very early in life, right, year 1 or 2 of their life. They were born with a few complications, pediatric hypertension, high blood pressure, very rare in children, and then a few other complications. And then when they were, I think, 15 months or so, they died. And the same for two brothers had identical fates. In this case, we run into a different sort of problem. So we find one character that's changed. So this is the length of the gene. Imagine there's a particular gene here. This gene is very fundamental to our development. All the development of our organs, to create our organs, cells have to move around, etc., and lots of things have to happen. So all of that happens from this gene. So it's pretty fundamental gene, and what we find here is one character change out here, And this character has never been seen ever in any other person. It's never been seen, noticed at all. And the gene looks like it's a good explanation for the disease. However, the character change that we see in the patients at hand is something that's never been seen before. Not only has it never been seen before, all the other character changes that have been seen are all in a different part of the gene. There's very little in this portion of the gene that's ever been seen. And we run into this problem repeatedly. And now you're in the complete realm of the unknown. You think you have a candidate, and it's an important decision because the mother of these two children is going to have her next baby soon, and they're going to decide whether to abort that baby or not, depending upon what you find here. So that, there are lives at stake here. And so now here is a character about which you know nothing at all, and you've got to make that decision. So those are the sorts of problems you run into. So how do you make these decisions? There are a variety of tricks, and none of them fully convincing other than eventually going to an experiment, but you do build up confidence using a variety of tricks. One of those tricks is to do the so-called conservation. We've never seen this variant in humans before, but has it been seen in any other animal before? Has it been seen down 500 million years or a billion years of evolution? Has this character ever been seen? So you look at all kinds of animals and say, at that position, what do you see? Do you see a departure from the norm? And in this case, as you can see, everything is a D. There's no departure from the norm. So like 500 million years of evolution, if you trace all of these organisms back, that character hasn't changed at all. And you can go another 100 million years back by looking at a family of genes, including that gene that we talked about, which diverged about 550 million years back. And even there, that character is completely preserved. It looks like over 500, 600 million years of evolution, there's been no departure from that character. And these two children have a departure from that character. And now, can we use that as a basis for abortion, etc.? Right? Of course, this leads to a nice machine learning problem that everybody works on. Given a character, can you predict whether it's problematic or not? And what sorts of features do you need? And so there's a whole literature on that. I have only 10 minutes, so I'll just continue on these cases, which is more interesting probably than the results of that machine learning expedition. We'll give you some accuracy, which is relatively poor, and so you don't know what to do with it, but there's a lot of scope for research going forward. Then you have the same children actually had another confounding variant where an A here became a G here. Now, what is interesting about this variant is it occurs right at the start of the gene. Yeah? So this is the start indicator. Just as gene recipes have end indicators, end of recipe indicator, which is an X, they have start indicators, which is an ATG. So ATG signals the start of a gene. If the ATG becomes a G, then the start has changed. So now maybe the gene doesn't know where to begin. Maybe our cells don't know how to execute this recipe because they don't know where to begin. So is that a serious problem or not? So now how do you make your way forward in such situations? So it so turns out that when such things happen where the start changes, our cells continue looking for the next possible start that's out there. Right? So then you say, okay, let me write down what property the start should satisfy. And so you take all the starts of all the genes and align them and say, okay, it looks like there's a pattern of this sort. The start has to be an ATG, but next to that there's likely to be a C, but maybe a G. 2 characters away, there's likely to be an A or a G, definitely not a T. So there's some rough pattern that emerges. And then you use that pattern to say, let's take the next 6 starts, potential ATGs in the gene, and say what does the pattern around those look like and which of those satisfies this pattern the best. And then you make a guess saying, look, maybe this one satisfies the pattern the best. And so now that's where the gene begins. And now you do all your predictions based on that point onwards. Maybe you're right, maybe you're wrong. So these guesses have to be taken, and maybe one needs better prediction mechanisms to see which is the real next start. Because the next start is a competition between who's first in line and who fits this pattern the most. So it's a 2-dimensional sort of problem. And so the first guy who fits it best will become the start, but the first guy who doesn't fit it well may give way to the second guy who fits it better, and so on. So those sorts of problems arise. And then occasionally we have these real conundrums where, here was a case of heterotaxy. Heterotaxy is when the organs are out of place. So typically our stomach is on our left and liver is on our right, and sometimes they get flipped. And that causes various problems. And when we looked at that family, there were 2 children with that problem. So the children looked like this. The father looked like this and the mother looked like this. So let's see what the children have. This character deleted, this T deleted in all of their gene copies. The father has it in one of his genome copies, so clearly he's provided that one copy to his children, both his children, but the mother has nothing at all. The mother is perfectly fine. If the mother is perfectly fine, then how did the children have this deletion in both their copies? What is the mystery here? So you run up against mysteries like this. And what could be an explanation for this? The explanation turns out to be very interesting. The explanation turns out to be eventually, and this we had to really work hard on this, the mother actually has a huge deletion in her one copy of her genome. Because she has a huge deletion, that is inherited by the children as well. And this copy of the genome provides no pieces at all. When we chop the genome into pieces, you've got to get pieces from somewhere. If there's nothing out here, you'll not get any pieces from here. All the pieces will come from here. And they'll give you a feel that both copies of the genome have that deletion, when really only one copy has it. The other copy has nothing at all. It has a much larger deletion. Much larger than any of those pieces, so you can't find it based on those pieces alone. Right? So these huge deletions happen sometimes, and for these you need special algorithms called split read algorithms, where you have to take a read and split it into two pieces. Because the only way to find these guys, just by searching for the reads, you won't find it. You have to take a read, break it into two pieces, and search for it independently. And the reason for that is when you search for these reads, you allow for a certain number of insertions, deletions, and errors. And you keep that number to a relatively small number. For those of you who do algorithms, you'll realize that the larger the number, dynamic programming goes for a toss. So you keep it to a small number. But when this is a huge deletion, that small number basically gives you nothing. So either you have to run dynamic programming with a huge gap parameter, or you just split the read into two pieces and do each piece independently. But then you know when you need to know where to split, and that's a little bit of trickery there. You sort of do the dynamic programming and until you get stuck, and then you say I'm going to split it there and then take the remaining piece out and so on. So small tricks like that. And so we find a number of these cases where you have these split reads that go far away and there's huge deletions that they encompass. Okay, I have time for two more cases. Now, we talked about introns and exons. Remember, the recipe is written in the exons and not in the introns. So the triplets happen in the exons. In between are introns, which I just skipped over. There's nothing out there. Okay. Now, occasionally you run into a case. Beta thalassemia is a very common disease in India. 1 in 25 people is a carrier of the disease. So they have the mutation in one copy of the gene. Luckily, that doesn't do much. But when it happens in both copies, it creates a problem. So 1 in 25, literally, there'll be at least one carrier in this room, likely. So in this case, we scratched our heads and found nothing in the exons. So the recipe seemed untouched. But in the introns, there was this character, this T had become a G, deep inside the introns. So the question was, okay, what could this do? Could this create a problem? And it turns out that introns, even though they're not important, they don't carry the recipe, our cells still have to figure out how to jump across from the one exon to another. And that jumping is quite problematic. The way that jumping happens is there are end markers here. There's a GT at the end of an exon and AG at the beginning of the exon. So you jump from here to here. Okay, so these GTs and AGs signal the ends and beginnings of exons. But then these are two character motifs, so they'll appear all over the place, right? So there'll be plenty of GTs and AGs in between as well. So there must be more signals for you to figure out how to jump from here and land there. So there are indeed a lot more signals. There's something called a branch point, which is a single character A. And then between the A and the AG, there has to be a stretch of characters, which is primarily composed of C's and T's. There might be a couple of A's in there or G's in there, but primarily it's C's and T's. So from here, you jump to the A, and from there, you jump to the AG, and the distance between this and this should not be too much. And so there's a whole bunch of rules that simply by studying these sequences across genes, people have inferred. Okay, so that being the case, let's go back to our T to G character change. What did it do? So somewhere here, a T had become a G. So what did it do? So the jump happens to the branch point here to the A, and from there to the AG at the end. So once you come to the A, it jumps to the AG in the next possible AG. What happened here was, unfortunately, this T became a G and just happened to be next to an A, sheer chance. So you got an AG coming right in between out here instead of that AG. And so it started jumping here instead of there, and so the exon began from here onwards instead of from there onwards. So you get 11 more characters in the recipe, 11, unfortunately, not a multiple of 3, so it creates a problem, right? So, and that caused the whole protein to go haywire. Okay, so let's jump to the last case. I have 3 minutes left. So this is a very interesting case. This is color blindness. So I don't know how many of you have taken these tests where you're shown the circle with dots in it and asked to call out the number that is hidden there. So when I was doing my entrance, entering IIT, they took a medical test, and so I did that test and found that I was red-green colorblind. So that was news to me. I'd never realized that. And it let it live for some time till about 3 years ago, when finally I could sequence my genome and look at it and said, let me figure out why I'm red-green colorblind. And so the cause happens to be very, very interesting, so we'll see. So how redness and greenness is perceived is that you have a red gene that creates a protein that is able to detect red wavelengths, let's say. And you have a green gene that creates a protein that detects green wavelengths. And then you have a blue gene, and the blue gene is somewhere far away, is not relevant to a story. The interesting thing is that the red and green genes are very similar to each other. It's practically as if they evolved, separated only very recently in evolution. So before that, they were together one. So mice, for instance, can see only 2 colors, can't see 3. So in humans, this separation has happened. And interestingly, there are 2 copies of the green gene, one here and next to each other. And this one doesn't do anything, so it's just lying around in the genome. So this red gene and this green gene are instrumental. So when I sequence my genome, I'll get a lot of these small snippets, and when I go and search for them, I lay them out like this. Nothing lands up here because this is identical, so I have to put each of these guys somewhere. I can put them here or here, it's the same thing. So as a convention, I put them all out here. And similarly, the 1st and 6th exon of the green and the red genes are identical. So I don't, I can't differentiate between them. So ignore the 1st and the 6th exons, okay? So the 2nd, 3rd, 4th, and 5th exons are what are important. Okay. So now, when you have 2 genes, very similar genes next to each other, what happens during meiosis is something called crossing over. New products get created where you take, this is 1 copy of the genome, the first one, this is the 2nd copy of the genome, each of us has 2 copies. You take this copy and this, you take from, until here you take this copy, from here onwards you take this copy, so the black bars, join them together, and that's what you pass on to your children. You don't pass on 1 copy by itself, you pass on a mosaic. When something like this happens, you get a mosaic getting created, and sometimes this process goes bad. It starts doing things like this. The cut happens here and here. So this part gets joined with that part. And that happens only because these 2 genes are very similar to each other in sequence. The red and green are very similar, so the similarity in sequence fools the cell into doing this mosaic by cutting here and here, rather than cutting at the same place in both chromosomes. When that happens, you start getting these hybrid genes, right? Imagine what happens here. You get 3 exons of the red, 3 of the green. So you start getting these sorts of situations, these sorts of hybrids, where you have genes like this, 3 greens and 3 reds, 3 greens and 3 reds, and so on. So by counting these reads appropriately and so on, you can get some numbers out here, which essentially allowed me to detect that this is my configuration out here. I have 3 greens and 3, I have a full red gene, but my green gene is half green and half red. So my red and green detection frequencies have come closer to each other, which means the ability to distinguish between the 2 has reduced. And so I'm a little more red-green colorblind than most of us are. So that's the talk, I think, 0000. That's great. So all of these cases are just finishing a book that captures all of these cases in case you are interested. It's on my website and you can download it. Thank you.